These classes about the minerals, copper, fluoride, and selenium. We will see one by one. First, we will see about the trace element copper. It is considered to be an essential trace element. What do you mean by trace element? If the recommend is less than 100 milligram per day, it is called as a trace element. This copper acts as a cofactor for many of the enzymes. It is present in the biological system as Cu plus and Cu2 plus valence state. That is, it is present in the cuprous and the cupric form. Coming to the dietary recommend of copper, adult requires about 1.5 to 3 milligram per day according to Food and Nutritional Board. Infants and children, they need 0.5 to 2 milligram per day. So, if you see the major dietary sources of copper, copper is spread over in these foodstuffs like cereals, meat, liver, nuts, and green leafy vegetables. And milk is considered to be a very poor source of copper. From the food, copper is partially absorbed in the stomach. So, stomach has a highly acidic environment, right? So, this acid environment, they release the bound copper ions from the partially digested food particles. And larger portion of the ingested copper, they enter into the duodenum and the ileum. So, duodenum and the ileum are the major sites of copper absorption. This copper is complexed with the amino acids, organic acids, or other chelators. A high fraction of the copper is soluble in the intestine tract. So, within the intestine, how it enters from the lumen to the cytosol of the enterocytes. It needs two transporters. One is a divalent metal transporter. The second recently identified transporter is a CTR1, which is a homotrimeric protein, which is conserved from the yeast to the humans. They transport the copper across the lumen, from the lumen to the cytosol of the enterocytes with high specificity and affinity. In the enterocyte, it has two phases, whether it can be stored along with the protein, smaller protein called as metallothionein, or it can be functionally utilized. We'll see about the metallothionein. Metallothionein are the group of small protein which has a molecular weight of 6.5 kilodaltons. These proteins, they are present in the liver, kidney, and intestine. So these metallothionein have high cysteine content. Cysteine has an SH group, right? So this SH bind with the copper, zinc, cadmium, and mercury compound. Why it has to be stored along with the metallothionein? Because when it is present in the free form, they generate the free radical which causes toxic damage to the enterocytes. So in order to keep it in the non-toxic form, they bound with a protein called as metallothionein. So these metallothionein level will be elevated when there is acute intake of these metals. For example, in case of copper poisoning, the level of metallothionein is going to get elevated. Also, when there is administration of hormones and cytokines, so these proteins are elevated. So metallothionein are a group of small proteins. They have high cysteine content. The SH group of the cysteine of the metallothionein, they bind the copper. The second fate is it can be functionally utilized, right? So it has its many functions. So copper is targeted to the proteins or organelles by a specific copper chaperone proteins. So copper is delivered to the mitochondrial cytochrome oxidase by a chaperone called as COX-17. It is targeted to superoxide dismutase via the chaperone called as copper chaperone superoxide dismutase, CCS. So also it is targeted to the via the ATOX1 to the cytosolic copper binding domain, of copper transporting ATPases, ATP7A and 7B. These two proteins are considered to be very important in the metabolism of copper. So where do these proteins are localized? So these ATP7A and 7B, they are predominantly localized in the trans-Golgi network, otherwise known as TGN. TGN is a trans-Golgi network. So these ATP7A and 7B proteins, they are localized across these trans-Golgi network. The function of ATP7A is the efflux of the copper from the enterocytes into the portal circulation is mediated via this protein. So the function of ATP7A is copper from the enterocyte is exported into the portal circulation by a protein called as ATP7A, which is localized in the trans-Golgi network. So from the food, copper is entered into the enterocyte via the divalent metal transporter and CTR1. From the enterocyte, they reach the portal circulation across a basolateral membrane through the protein called as ATP7A. From the portal circulation, it reaches the liver and it is transferred and further distributed through the bloodstream throughout the entire organ, which includes the muscles and the brain. So within the hepatocyte, how it crosses the basolateral surface of the hepatocyte. So the protein ATP7B, they pump the copper out of the hepatocytes. So the cellular trafficking of the ATP7A and 7B is reversible. So ATP7A is present in the enterocytes and ATP7B is localized in the hepatocytes. So whenever there is a cellular concentration of copper is increased, these proteins ATP7A and 7B, they are redistributed to the post-Golgi vesicles or to a cell membrane to facilitate the cellular copper export. When the copper level is decreased, what happens? The transporters, they are recycled back to the trans-Golgi network. So the cellular trafficking of ATP7A and 7B is reversible. So from the food, it reaches the duodenum and the ileum. It is absorbed across the lumen into the cytosol of the enterocytes. From the enterocytes, it reaches the portal circulation. From the portal circulation, reaches the liver and further distributed across the other organs. So in the serum, how it is present? It is approximately 90% of the copper, they get bound to the copper binding protein called a ceruloplasmin. These ceruloplasmin, they are synthesized within the hepatocytes. It is a 160 kilodalton protein. It is an alpha-2 globulin protein and each atom can bind with, you can see 6 copper atoms, right? So every molecule of ceruloplasmin can bind with the 6 atoms of copper. So the color of the ceruloplasmin is blue because it has a high content of copper. So ceruloplasmin is synthesized from the liver and each atom of ceruloplasmin can bind with the 6 atoms of copper. So this ceruloplasmin has a ferro-oxidase activity, which means the iron atom from the ferrous state is oxidized to the ferric form and further it is incorporated into the transferrin. So this ceruloplasmin level will be reduced in case of liver disorders. Also, in addition to ceruloplasmin, some amount of copper is loosely bound to the albumin, which is considered to be more important because of its loose binding ability. They readily release the copper to the tissues. And also, copper can bind with the transcriptrin. So it binds with mostly with the ceruloplasmin, which is a tight binding, and loosely with the albumin, and also it binds with one more protein called as transcriptrin. Why it has to get transported along with the transport protein? Because copper in its free form, what happens? It has high redox activity and they generate the free radicals. They oxidize the proteins, lipids and nucleic acid and they generate the free radicals. In order to prevent the uncontrolled redox activity of copper under physiological condition, this copper is almost exclusively associated with the transport protein and its free level has to be maintained at a very low level. That is, it should be less than 10⁻⁻8 moles per liter. So this amount is less than 1 free copper ion per cell. So it has to be bound always with the transport proteins. Whenever there is an increased concentration of the free ion, which means it generates a free radical and causes toxic damage to the macromolecules like lipids, proteins and nucleic acid. So the normal blood level of copper is 100 microgram per deciliter and total body content is 100 milligram. So the copper is mostly distributed in the liver, brain, kidney, heart, bone, muscle and hair. So how it is excreted? So 0.5 to 2 milligram per day of copper is excreted via the bile into the feces, and urine copper output is less than 60 microgram per day. So copper in the food get absorbed in the mostly in the duodenum and ileum. In the enterocytes, it has two transporters, divalent metal transporter and CTR1. It transports copper into the intestinal epithelial cells. It has two fates. One, it can be stored along with the metallothionein, or it can be functionally utilized. So using the ATP7A protein, it enters into the circulation, that is portal circulation, reaches the liver and redistributed to the most of the organs. So, and it gets excreted via the, mostly via the bile into the feces and some amount in the urine. Next, we will see about the biochemical functions of copper. Copper acts as a cofactor for many of the enzymes. So first is the cytochrome C oxidase. This cytochrome C oxidase is involved in cellular respiration. Also, it is a cofactor for ceruloplasmin. You know that ceruloplasmin has a ferrooxidase activity. It is called as ferrooxidase 1, right? So it oxidizes the ferrous ion to the ferric ion, and it incorporates the ferric ion into the transferrin. So thus it is involved in the ion homeostasis. Also, it is needed for the formation of hemoglobin, myoglobin, and cytochromes. So it is a cofactor for ALA synthase. ALA synthase is a rate-limiting enzyme in the hemoglobin biosynthesis. Also, it is a cofactor for tyrosinase. This tyrosinase enzyme is involved in melanin synthesis. Copper is also a cofactor for various enzymes involved in neurotransmitter production. It is also a cofactor for lysyl oxidase. Lysyl oxidase is involved in the synthesis of connective tissues. This lysyl oxidase oxidizes 4 lysin residues to form a desmosin compound. This desmosin, they cross-link with the elastin and stabilizes the extracellular matrix. So this enzyme is associated with the, where the distribution is, it is present in the connective tissue located in the aorta, dermal connective tissue, and in the fibroblast. Also, it is a cofactor for ascorbic acid oxidase, which is involved in cross-linking of collagen molecule. And it's a cofactor for superoxide dismutase. They protect against the oxidants. And also copper accepts and donates the electron involving oxygenation, hydroxylation, and dismutation reaction. Also, it is a cofactor for desaturase enzyme. This desaturase is an enzyme which is involved in the addition of double bonds in the long chain fatty acids. Also, copper is a cofactor for uricase enzyme, which is involved in degradation of uric acid. Till now, we have seen about the enzyme proteins. The copper acts as a cofactor for various enzymes. Next, we will see about the copper containing non-enzymic proteins. They are hepatocuprin, which is a storage form in the liver. Hepato means liver, cuprin means the copper. So hepatocuprin in the liver, cerebrocuprin in the brain, erythrocuprin in the bone marrow, hemocuprin in the RBC, and hemocyanin, which is present in the crustaceans. They are the oxygen-carrying blue pigment. Next, we'll see about the normal levels of copper. Serum copper level should be 10 to 22 micromoles per liter. And ceruloplasmin level should be 20 to 60 milligram per deciliter. And urinary copper, it's less than 1 micromoles per liter per day. That is always urine level should be expressed as day, right? So urine copper, it is less than 1 micromoles per day, and liver copper content, it should be expressed as a dry weight of liver. So the normal liver copper content is 20 to 50 micrograms per gram of dry weight. So I'll repeat it once again. The normal level of serum copper is 10 to 22 micromoles per liter. Ceruloplasmin is 20 to 60 milligrams per deciliter, otherwise 200 to 600 milligrams per liter, and urinary copper is less than 1 micromoles per day, and liver copper, it is 20 to 50 micrograms per gram of dry weight of liver. Next, we'll see about the disorders associated with the copper metabolism. One is the Wilson's disease. Second thing is the Menkes-Pinky syndrome. Third is a aceruloplasminemia. Fourth one is a copper deficiency anemia. The anemia will be microcytic normochromic anemia. And copper deficiency may also lead to weakening of the vessel wall, which results in aneurysm and a fatal rupture, and hypopigmentation and ataxia. We will see one by one. First, about the Wilson's disease. It's a disorder of copper metabolism. The incidence of Wilson's disease is 1 in 50,000. It is transmitted as an autosomal recessive triad. So the defect here is, what is the pathology here is, there is copper binding ATPase is defective. That is a mutation in the gene coding for copper binding ATPase 7B. So what the function of ATP 7B is, it's help in the export of copper from the liver, right? So the excretion of the copper from the liver is affected, and hence there is accumulation of the copper in the liver and brain. So this condition is called as hepatolenticular degeneration. The level of ceruloplasmin is reduced. So normally we know that after its absorption in the intestine, it enters a portal circulation, reaches the liver. In the hepatocyte, it stimulates ATP7B protein, which has two major functions. One is it binds the copper to the apoceruloplasmin to form ceruloplasmin. The ceruloplasmin is a copper carrying protein. The second function of ATP7B is it packages the copper into a vesicles for exocytosis into the bile. So the ATP7B has two major functions. One is it binds the copper to the apoceruloplasmin to form ceruloplasmin. This ceruloplasmin is a copper carrying protein. The second function of ATP7B is it packages the copper into a vesicles so that it gets exocytosed into the bile. In case of Wilson's disease, what happens? The defect here in the excretion of the copper, right? So ATP7B will be defective. What happens here? There is an accumulation of the copper within the hepatocytes. Copper cannot bind with the apoceruloplasmin to form ceruloplasmin because ATP7B is defective. There is accumulation of copper within the hepatocytes. The free copper, what happens? If it is present in excess amount, they bind with the hydrogen peroxide to produce a free radical. This free radical is toxic to the hepatocytes and causes destruction of the hepatocytes. This hence the copper is released into the circulation. So it reaches the various tissues and organs. So the copper deposit in the basal ganglia causes the movement disorder, otherwise known as ataxia, and the deposition of the copper in the cerebral cortex causes dementia. In the eye, there is a deposition of the copper, that is, it produces a golden green pigment ring around the cornea due to the deposition of the copper in the Descemet's membrane. So the copper gets deposited in the Descemet's membrane of the cornea to produce a green or golden color pigment ring called as Kaeser-Fleischer ring, otherwise known as, we would say it as KF ring. So the KF ring is the deposition of the copper in the Descemet's membrane of the cornea. Also, the free copper is toxic to the hepatocytes, will induce the acute hepatitis. In a chronic course, it causes cirrhosis. The symptoms develop in the late childhood period. The two factors you need to consider in Wilson's disease is in the blood, there will be decreased ceruloplasmin level. Normally, ATP7B is responsible for the binding of the copper with the apoceruloplasmin to form the ceruloplasmin, right? So in Wilson's disease, ATP7B is defective and hence copper cannot bind with the ceruloplasmin, and this ceruloplasmin is unstable in the plasma and hence the level of the ceruloplasmin will be reduced. Okay. Next is the level of the copper in the serum. The free copper level will be elevated. So there is increased accumulation of the copper. The copper causes toxic damage to the hepatocytes by forming a free radicals. This free radical damages the hepatocytes. It gets entered into the circulation and reaches the various tissues and organs. So in the systemic circulation, if you assess the free copper level, the level will be elevated. Also, it gets spilled in the urine and hence the level of copper in the urine is also elevated. The other complications in Wilson's disease are enlarged liver and spleen. Enlarged means megaly, right? So liver and spleen, it is hepatosplenomegaly. So it also causes renal disorders. The free copper damages the proximal tubular epithelial cells and causes the renal damage. Also, the free copper damages the red cell membrane and hence causing the hemolytic anemia. If you suspect a case of Wilson's disease, you need to do two things. One is the liver biopsy. The liver copper content over 250 microgram per gram of dry weight of liver, and whenever the plasma level of the ceruloplasmin is less than 20 milligram per deciliter, these two things are considered to be diagnostic if you suspect a case of Wilson's disease. So the liver biopsy, if the content is more than 250 microgram per gram of dry weight of liver and the plasma level of the ceruloplasmin is reduced, that is less than 20 milligram per deciliter, is diagnostic of Wilson's disease. What is the treatment for this condition? Administered penicillamine. Penicillamine is not an antibiotic. It is a copper chelating agent. It chelates the copper and gets excreted in the urine. Also, zinc along with ammonium tetrathiomolybdate, they decreases the copper reabsorption and hence it is excreted in the urine. And the last resort is if the patient develops cirrhosis and liver failure, you need to do the liver transplant. So there are three modalities of treatment. One is the penicillamine. It chelates the copper and gets excreted in the urine. Administration of zinc and ammonium tetrathiomolybdate. This causes decreased copper resorption and hence the copper gets excreted in the urine. The last and final resort is a liver transplantation. The second disease is the Menkes disease. In the Wilson's disease, there is a defect in the ATP7B gene, right? In case of Menkes disease, there is a defect in the ATP7A gene. It is transmitted as an X-linked recessive triad, which means that male child is going to get affected more, right? So this protein is observed by the copper. This protein is involved in the transport of the copper. That is, it exports the copper from the enterocytes to the portal circulation. So copper is observed by the intestinal cells, but the observed copper cannot enter into the blood. So copper gets accumulated in the intestine and in the variety of other cells and tissues. This disease involves the nervous tissue, connective tissue, and vasculature. So the copper is observed in the intestine and it is not exported out from the intestine to reach the various tissues. And mainly involved organs are nervous tissue, connective tissue, and the vasculature. Here there is no liver involvement. So the activity of the copper dependent enzyme is going to be decreased. So the level of copper is decreased, right? So the activity of copper dependent enzyme is going to be decreased. And the patient will be having a poor prognosis and the child usually dies during infant period. So early diagnosis is important in this case because treatment with copper is going to be effective for these patients. So assess the serum copper level. The serum copper level is reduced in case of Menkes disease. The third condition is aceruloplasminemia. It is also a genetic disorder. Congenitally, there will be low level of ceruloplasmin. You know that ceruloplasmin has a ferrooxidase activity. If there is if the ceruloplasmin level is low, so deficient ferrooxidase activity, and this causes failure of the release of the iron from the cells, and hence iron gets accumulated in the brain, hepatocytes, and pancreatic islets, and neurological symptoms are seen in this patient. The fourth defect in copper metabolism is copper deficiency anemia. Copper is essential in the formation of hemoglobin. The first thing is copper containing ceruloplasmin, they maintain the iron homeostasis, right? So the second is it is an integral part of the ALA synthase enzyme. ALA synthase is a rate limiting enzyme in hemoglobin synthesis. Also, copper is needed for the uptake of iron by the normoblast. Hence, the patient presents with microcytic normochromic anemia. The size of the RBC is reduced. It has to be compared with the small lymphocytes. So RBC is the size of the RBC is 7 microns and small lymphocyte is also 7 microns. So it is microcytic normochromic anemia in case of copper deficiency. If there is added iron deficiency, microcytic hypochromic anemia will be developed. Next is the cardiovascular disorders. Copper is a constituent of lysyl oxidase, you know, right? So this lysyl oxidase oxidizes 4 lysyl residues to form a desmosal which cross-links with the elastin and stabilizes the extracellular matrix. In case of copper deficiency, what happens? This elastin becomes abnormal and hence there is weakening of the walls of the major arteries. Because of the weakening, there will be development of aneurysm and there will be fatal rupture of the wall of the aorta. Also, the patient will be having the fibrosis of the myocardium leading to cardiac failure. Also, copper is needed for melanin synthesis. Copper is present in tyrosinase. This tyrosinase enzyme is needed for melanin synthesis. Hence, in case of copper deficiency, the patient presents with hypopigmentation and graying of hair. In the period of deficiency, it is marked by the alternate white patches regarded as the flat type hair growth. Till now, we have seen about the deficiency of copper. Next is about the copper toxicity. So what happens if there is excess copper? If it is present in the free form, what happens? It oxidizes the proteins, lipids, and nucleic acid, and they increase the free radical production. So chronic copper toxicity, they lead to diarrhea and bluish-green discoloration of the saliva. In the circulation, if the copper content, free copper is present, it causes damage to the reticular RBCs, and this causes hemolysis, hemoglobinuria. Also, there will be proteinuria and renal failure. So, if there is copper toxicity, what happens? There will be increased in the synthesis of the metallothionein. Metallothionein is a small group of proteins present in the cytosol of the liver, kidney, and intestine. It has a high cysteine content, and the SH group of the cysteine, they bind with the copper, zinc, cadmium, and mercury. Till now, we have seen about the copper and its metabolism, its deficiency states, and its toxicity. Next, we will see about the another trace element called as fluorine. What is the source of fluorine? The sources of fluorine are water from the deep subsoil wells and irrigation dams and high fluoride content. They are present in the sea fish, tea, cheese, and java. Also, the fluorinated toothpaste contains a fluoride content of more than 3000 parts per million. Also, the ordinary toothpaste contains 700 parts per million. What is parts per million is? The parts per million is 1 gram of fluorine in million gram of water. It equals to 1 milligram per liter. 1 milligram of fluorine in 1 liter of water is 1 parts per million in water. So the normal level of fluorine in blood is 4 microgram per deciliter. The safe limit of fluorine is 1 parts per million in water. What happens if there is a pit and fissures in the premolar and the molar tooth? There will be accumulation of the food particles in the pits and fissures, and the bacteria will ferment the food particles. There will be bacterial fermentation. There is a formation of the acid, right? So this acid will remove the enamel. So in this picture, this is the enamel, this is the dentine, this is the pulp of the tooth. So this is the enamel, this is the dentine, and this is the pulp of the tooth. What happens when there is bacterial fermentation is, it is exposed to the acid. The enamel is exposed to the acid and it removes the dentine and the enamel, exposing the pulp, and hence leads to inflammation and toothache. What is the treatment for this case? Is topical application of the fluorine. They provide the fluoroapatite, that is a formation of fluoroapatite layer on the enamel, and hence it protects it. This fluoride ion, how it forms the fluoroapatite crystal is, they enter into the hydration shell surrounding the apatite crystals and they get incorporated into the crystal surface. Thus makes the tooth surface more resistant to plaque bacterial attack. So the safe level is 1 parts per million. If it is more than 2 parts per million, the patient will be having chronic intestinal upset, gastroenteritis, loss of appetite, and loss of weight. If it is more than 5 parts per million, the patient will be having a mottled enamel along with stratification and discoloration of teeth. If it is more than 20 parts per million, it is toxic. It causes skeletal fluorosis. There will be alternate areas of osteoporosis decreased bone density, and osteosclerosis, increased bone density. So osteoporosis alternate with an areas of osteosclerosis, thus causing brittling of the bone. And the patient presents with genu valgum. There are so many causes of genu valgum, and one cause of genu valgum is the fluorosis. The patient will be having a knock knees. If the patient is asked to stand straight along with the knees together, there will be large gapping in the ankles, right? So this is called as the knock knee. How are we going to prevent the fluorosis? That is, provide the fluoride-free water to the population, and restriction of the intake of jowar, and supplementation of the vitamin C, regulation of the fluoride-containing toothpaste. Next, we'll see about the another trace element, which is called as selenium. What is the source of selenium is the food crops grown in selenium-rich soil is a source, and the requirement of selenium is 50 to 100 microgram per day. And the serum selenium content is 50 to 100 microgram per deciliter. And the highest concentration of the selenium is present in the testes. This selenium is needed for the normal development of spermatozoa, and it is concentrated in the midpiece of spermatozoa as a specific selenoprotein. There are variety of enzymes which are selenium dependent. We'll see only three. One is the thioredoxin reductase. Next one is the glutathione peroxidase, which is present in a higher concentration in the RBC, and deiodinase enzyme. So these three enzymes are the important selenium containing enzyme. One is the thioredoxin reductase, glutathione peroxidase, and deiodinase. In case of selenium deficiency, there will be defective deiodinase activity, and hence the patient presents with hypothyroidism. T4 cannot be converted to T3, and hence the patient presents with the hypothyroidism. Also, there is a presence of selenoprotein, which is an antioxidant present in the blood, and it contains about 16 residues of selenocysteine. You know that selenocysteine is a 21st amino acid, and it is coded by the UGA stop codon, and this selenocysteine is incorporated into the protein during the biosynthesis. We'll see about the selenium deficiency. The patient will be having, the condition is called as the Cation's disease, and the patients will be having the multifocal myocardial necrosis, cardiac arrhythmia, cardiac enlargement, liver necrosis, cirrhosis, and muscular dystrophy. If there is selenium toxicity, the patient will be experiencing hair loss, fall of nails, diarrhea, weight loss, and garlic odor in the breath. Thank you.
These are my references.